Thank you for the introduction. So I'm happy to present our first experiences with whole genome sequencing, which is an example how whole genome sequencing can be used in the food industry. Before going to that, I will give a brief overview of analytics which are done internally within Nestlé. So within Nestlé worldwide, we have different facilities where analytical services can be provided. And this is to support corporate quality management and to assure the quality of Nestlé products. So we have two research centra which are located in Lausanne in Switzerland. And then we have 39 regional R&D centers where some analytical work can be done. For the more routine analysis in microbiology and chemistry, there are 25 Nestlé Quality Assurance centers. And then there are another 45 labs who are doing limited analysis, which are associated with factories. So you see here a distribution of the Nestlé Quality Assurance Centers where the routine analysis are done, which are located, in fact, all over the world. To give an idea how many tests we do in microbiology annually within Nestlé, it's around 5 to 6 million tests per year. And this is not including the analysis which are outsourced, so just internally. The purpose of analysis is, in fact, the release of raw materials, finished products in the first place, and then followed by monitoring and surveillance studies or other investigations. So, in fact, identification and typing is a really important activity within the food industry. It supports investigations. It is used for factory ecology studies, factory issues, lab issues, and also supplier follow-up. And depending on the level of information that we needed, we use the right methodology, and most of the times at that level it will be a molecular assay. And we are very interested in whole genome sequencing for source tracking analysis because of the high discriminatory power that it has and the decrease in cost during the recent years. So before going to elaborate on two case studies where we used whole genome sequencing, I would give a bit of time on how whole genome sequencing, the methodology which was used in our hands. So in the first place, what is done for the different isolates, we will grow them so that we have a pure culture, and from there we do a DNA extraction. This DNA is then sequenced by an Illumina MySeq run, and then this sequence data is then pre-processed so that we retain only the high-quality raw sequence data. So then the data is ready for bioinformatic analysis, and for this we use a Snip approach. So in the previous presentations this was a bit clarified what a Snip is, so I will not explain that. But let us say for the Snip workflow, what we need is a reference. So this is a closed genome that is used to map all the raw sequence data as a kind of a scaffold to map all the raw sequence data of all the isolates. And then at every position at the genome, it is looked whether there is a Snip, yes or no. And for the Snip, you have a kind of a criteria that you need to choose on whether this is a Snip, yes or no. In our case, the Snip criteria was that you should have at every position at least 8 times depth, and then the nucleotide difference that is observed should be seen in 90% of the reads, let us say. Then of all the isolates, these SNPs are compared, and this is then used to build a phylogenetic tree. So the first case study where whole genome sequencing was used was as follows. So there was a food product which was sent to a Nestlé Quality Assurance Center, and that was a sample that was tested for a batch release. During the analysis, this food sample was found positive for Salmonella. So the next step, what is normally done, is serotyping, and apparently it was Salmonella hadar. But during, for a positive control in this lab for Salmonella testing was a Salmonella hadar as well. And then during environmental monitoring in the laboratory, there was a Salmonella found, which was also a Salmonella hadar. So the question here was, do all these isolates that we found here have a common ancestor? So this is an important question to answer. If the answer is yes, this means that the positive control contaminated the lab, contaminated the food sample, which means that the food sample was not representative for the batch. The food sample, the batch would be safe, let us say. But if the question is no, this means it's a sad coincidence that we had in the food sample a Salmonella hadar, which is the same serotype by coincidence than the positive control. Of course, we cannot decide with this level of information, and we needed a more detailed analysis. We evaluated ribotyping, as this is we can do it automatically. We use it for a long time already. And the ribotyping result gave that all the isolates of the food, the positive control, and the environmental sample had the same pattern. Of course, it is only looking at the ribosomal operon, which we know it's not really going up to strain level. So this analysis was not giving all the answers. And parallel, we did PFGE. And again, for all of the isolates, a same pattern of a PFGE pattern was found. It is known that you can find a same pattern for different types of strains. So we decided, let's do a whole genome sequencing. We applied the Snip approach, as explained before. And here you see the phylogenetic tree. So P1 and P2 are 2 times the positive control that were analyzed, E the environmental sample, F the food sample, and they were all clustering together. While S1 and S2, there were 2 Salmonella hadar strains that we had in our collection, and that we knew they were not related with the lab issue, let us say, and they were clustering separately. From this, and to show more into detail, what is the exact number of SNPs that we found, we show you this table. This is a pairwise SNP comparison matrix. So you see all the isolates again, P1, P2, etc. And you see the absolute number of SNPs that were found with the approach that we have taken between all of the isolates. The blue color, the intensity of the blue color indicates the high number of SNPs. So dark blue will mean you have a lot of SNPs, while white will be not so many SNPs. In this case, what we can see here, compared all the isolates to the reference, that the reference is quite close to the lab contamination issue or our issue that we are under investigation, and that the 2 isolates from our collection, they were rather far away from the reference. But the most importantly, if we look between the issue strains of Salmonella hadar, that less than 10 SNPs were found between them. So from there we concluded, okay, they are very close, they are having a common ancestor, and we are dealing with a lab contamination issue. We were a bit interested also to know, okay, if this Salmonella hadar, if you would grow them, how much genetic differentiation would you expect? And therefore we carried out 2 different independent experiments. In experiment A, it was you have a plate with Salmonella hadar, and we selected 4 colonies. And for each of these colonies, we extracted DNA and did the whole genome sequencing analysis. Then for the second experiment, experiment B, from one plate we took 5 colonies, and then from one of that colony we did 10 subcultures. And after 10 subcultures from that plate, again, we took 5 colonies and analyzed all these results by whole genome sequencing. What you can see here is the phylogenetic tree and the pairwise SNP comparison matrix. Again, here we can see that a maximum of 2 SNPs were seen between the 2 different experiments, which show that it's quite stable, and that assured us. Then the second case study that I would like to share with you, it's in fact a factory contamination issue. So what you see here on the slide, it's a part of a factory plan in a very simplified format. And where you see the white color is in fact the basic hygiene area, and the green color is the high hygienic area. Then if the product, within the production process, the product goes at a certain moment from a drying area, so this is in the high hygienic area, then go to the batch area where the product is divided into big batches. And then the big batches are then going to the filling area where the product is, let us say, packed in small packages. Here, what you can see is that at a certain point let us say, during the environmental monitoring, there was salmonella found in some of the environmental samples. It should be noted that there was no positive salmonella samples found within the finished product. And during this issue, the production process was directly stopped. What is in this slide added to the factory plan is the red boxes with the sample numbers. And these are the different sample locations where salmonella was analyzed. And in the table underneath the figure, you see again the different locations, and then the results of the salmonella analysis are presented. So the white boxes in the table means there was no salmonella found, while the red boxes, this means at certain days salmonella was found. We call it, to be simple, salmonella serotype 1. And then the orange is when salmonella serotype 2 was found. So for the salmonella serotype 1, we analyzed by whole genome sequencing. Why? Because the question was, okay, where is this salmonella coming from and how should we solve this factory issue? So we said, okay, we will like to know whether all the salmonella serotype 1 isolates have a common ancestor. Do they come from, if there is one source, or do we have different sources within the factory? So we analyzed all the isolates of salmonella serotype 1 together. And for this, we selected one strain that we sequenced by PacBio sequencing. Why? Because for a SNP analysis, we know that the reference genome is really important, that it's very close to the isolates to have a correct result. And then the PacBio sequencing allows you to have a complete genome, to create a complete genome, let us say, because you have long reads. So this was done, a PacBio, to have this complete genome. And then all the isolates were mapped against this reference. And here you see the results of the SNP analysis. What you can see is that a maximum of 4 SNPs were found between all of the isolates. And from that we can conclude that, okay, all the salmonella serotype 1 isolates have a common ancestor we are dealing with one source and at all the different positions, yeah, it's coming from one source, let us say. We did the same for the Salmonella serotype 2. So again, for this serotype, we did one PacBio to have a good closed genome that was used for reference. We did the Snip analysis, and from the table, we can see that a maximum of 2 SNPs were identified between all of the isolates. And again, we concluded also for this serotype, we are dealing with one source, and that the rest, all the isolates are spread within the factory. So this helped us in the investigation to solve the factory issue. So what in fact was done, that we were dealing with a kind of a forklift, which was, let us say, used, the same forklift was used through all of the area, and this means that there was a spread of the contamination everywhere. So what was done? Of course, the factory was cleaned thoroughly everywhere, but also the forklift wheels, and there was an additional action taken that outside this area of the figure that we see, that things which are coming from one forklift stop at a certain area and then give it to another forklift, and then so a kind of barriers and separation of forklifts from one area to another. And from that point, there was no Salmonella found anymore in the factory. What can we conclude from these case studies? That whole genome sequencing, it has a really discriminatory power and ability. You can reveal the sequence of the whole genome, which can allow you to go really into a detailed analysis up to strain level, which was really helpful in the lab contamination issue and the factory issue. The next step that we would consider is the validation of the whole genome sequencing. As we used it here, it is a source tracking tool. So we could imagine that we are going to look for how can we validate a typing tool. And an example of how this can be done is mentioned on the slide. Of course, what is another benefit, and that we did not discuss here, but we said only application for source tracking analysis, but you have the whole sequence of the genome. So you can imagine that you can retrieve much more information out of this. For example, if a strain is transient in a factory, and you would like to know, okay, why is the strain transient? Is there something in the genome that is different between other isolates which are publicly available? Why we see this strain and not another strain? So that can be very useful. From a food safety management perspective, whole genome sequencing is seen as really beneficial. Okay. It's really beneficial. Food safety management is really embracing this technology. Of course, the considerations are that food safety is global. This means that we need harmonization of whole genome sequencing protocols. As I showed in the introductory slides, we have labs all over the world. If one lab is doing an analysis and another lab is doing analysis, we would like to have equivalent results. And in this area, also proficiency testing is an important aspect. Also, as we presented whole genome sequencing here, it is used as a kind of a typing tool. And of course, epidemiological data, when used as a typing tool, stays indispensable. A hurdle of whole genome sequencing that is probably mostly the technical competence that is required between the different stakeholders. And we are talking about the industry, authorities, and so on, that we understand how the methodology is used and that everybody speaks the same language. Then another important point that might not be easy is the sharing of the data. We think it's important that sequence information is get out in a public database, like the genome tracker that was presented by Peter. We can learn a lot of it. So if you found isolates and you compare it with data in a public database, then you can see, well, the strain that we found, is it a new one? Is it already existing in the database? Or maybe it is closely related to strains in a specific area. So it will help us to find ways to find actions which are more specific and more targeted. And it will help us at the end. Authorities, food industry, everybody at the end. So this was my thoughts and our experience of whole genome sequencing, and I'm happy to hear your thoughts about this, and let's see. Thank you.